Welcome back to the second video in our study design series. In this video, we will introduce some important terms that will help you understand study designs. You may know some of these by now, but the first term we will talk about is bias. Bias is an error that results in an incorrect estimate of the true effect between an exposure and an outcome. Temporality is another term you'll hear a lot. It establishes a causal relationship when it is clear that the effect occurs after the cause. There are some study types in which this is not so clear, so we'll talk about that a little bit more. And confounding is the distortion of an association between an exposure and health outcome by a third variable, known as a confounder. Let's get into each of these a little bit more. There are some different types of bias you will learn about in Intro to Epi, and while you'll get more about this in class, basically it's important to understand that there are different factors that can influence the validity of a study. Two major sources of bias you will learn about are selection bias and information bias. These usually occur during the formation of a study, so it's important to be aware of the possibilities and maintain a strong study. Hill's criteria for causality is another important concept you will learn about in Intro to Epi. Essentially, it's a list of criteria that helps us determine the strength of a relationship between an exposure and an outcome. The more of these factors that are met, the stronger the relationship is considered to be. And temporality is really our main focus here, and it's number one on the list. It's the only one of these criteria that must be present in order for any causal relationship to be established between an exposure and an outcome. As we stated in the definition before, a relationship shows temporality when the effect comes after the cause. This seems obvious, but it's important to define because in some studies it's not always so clear. Some of the other criteria you'll see here include strength, which can be illustrated by a 2x2 table. Risk ratio greater than 2 is considered strong and usually means that the association is more likely causal. Really, a risk ratio further from 1 in either direction is significant, if you'll remember that. Consistency means that findings are replicated in more than one study, while specificity means that one exposure leads to only one outcome. An example of that would be infectious diseases. Biological gradient is also known as a dose response, which means the greater the exposure, the greater the probability or severity of the disease. And you'll see some others here, and you'll be responsible for identifying these during your time at Intro to Epi. So it's good to be familiar with each of them and be able to think about examples. Now to expand on confounding a bit, you'll recall this is when a third variable confuses the relationship between the two others. Here's a really basic example that you'll probably see at some point during the course. And when we observe the association between people who eat ice cream and those that develop a sunburn, sure, you could run a study and probably find a strong correlation between people who eat ice cream and get a sunburn. But there is an important third factor likely influencing this relationship. So the hot temperature is a confounder here because it is related to both of these things that we're studying and is confusing the relationship between eating ice cream and developing a sunburn. So that was a short video just to introduce a few concepts. We touched on these concepts important to research. We discussed bias, temporality, and confounding. Please continue to video number 3 where we'll start learning about the first study design, which is randomized controlled trials. Thank you.